Other and the density population also, no? The density. One gets one, they'll just get around.
Prabhu, just check on YouTube check பண்ணல பிரபு.
Sir?
பிரபு.
Yes sir.
YouTube check பண்ணு YouTube check பண்ணு கரெக்ட்டா வருதானோ? YouTube channel check பண்ணு.
YouTube
YouTube channel check பண்ணு கரெக்ட்டா வருதா?
வெபினார் சார் இன்னும் வரலையே. பண்ணிட்டீங்களா?
உபய் சார் ஆரம்பிச்சலாமா? யூடியூப் ஓகே ஆச்சா?
எவ்வளவு பேர் ஜாயின் பண்ணியிருக்காங்க?
69 சார்.
ஒரு 100 மேல ரெஜிஸ்டர் பண்ணிக்கா சொன்னீங்க.
359 நினைக்கிறேன் சார் ரெஜிஸ்ட்ரேஷன். டோட்டல் ரெஜிஸ்ட்ரேஷன் ஆகல சார். யூஸ்வலா போன வெபினார் அதெல்லாம் நிறைய ரெஜிஸ்ட்ரேஷன் இருந்துச்சு. இதுல 500 கம்மியா இப்பதான் நம்ம ஸ்டார்ட் பண்ணதுல இன்னைக்கு தான் 500 கம்மியா இருக்கு ரெஜிஸ்ட்ரேஷன். யூஸ்வலா 500 கிராஸ் ஆயிடும் மோஸ்ட்லி. ஃபர்ஸ்ட் பண்ணும்போது 2-3 வெபினார்ல வந்து 1000 தாண்டிடுச்சு. 2 வெபினார்ல 500 ஆச்சு. இந்த தடவை 500 கீழவே இருக்கு. ஆமா ஆமா அது வேற இருக்கு சார். இங்க சென்னையில இருக்கிறதுனால அது ஒரு இது நமக்கு. அப்போ மோஸ்ட்லி நம்ம ஸ்டூடெண்ட்ஸ் நிறைய பேர் நாம எக்ஸ்பெக்ட் பண்ணோம் சார். இன்னைக்கு சாட்டர்டே என்னால நிறைய பேர் ரெஜிஸ்டர் பண்ணி வருவாங்கன்னு நினைச்சோம். ஆனா நம்ம ஸ்டூடெண்ட்ஸ் நிறைய பேர் வரல. கம்மியா தான் இருக்காங்க நம்ம ஸ்டூடெண்ட்ஸ்ல. ஒரு கிளாஸ்ல ஒரு 4th செம்ல பாத்தீங்கன்னா ஒரு 5 பேர் தான் ரெஜிஸ்டர் பண்ணியிருக்கானுங்க.
விஜயகுமார் சார்
வரல.
ஹலோ விஜயகுமார் சார்.
எஸ் சார் ஸ்டார்ட் பண்ணலாமா?
4:30 registration. 4:30.
4:30 ஏன் அப்புறம் யார் அதுக்கு லிங்க்ல அனுப்பிட்டாங்களா?
நம்ம சிவகுமார் சொன்னாரு.
பார்க்கல, நேத்து பார்த்துட்டு தான் லாஸ்டா, அதுக்கப்புறம் பார்க்கல.
இல்ல காலைல
அனுப்பிச்சு யூடியூப்ல காட்டுது ஒரு 20க்கு மேல யூடியூப்ல.
மொபைல்
மியூட் போட்டுக்கோங்க.
மொபைல் மியூட் பண்ணுங்க சார். மொபைல அப்படியே சொல்லுங்க. Ma'am, shall we start, ma'am? Ma'am, good afternoon, ma'am. This is Mobile. Ma'am? Ma'am? Shall we start, ma'am?
Yes, sir. 1:59 ஆகுது.
Yeah, 2 o'clock sharp you start.
Okay. A very good afternoon, all of you. Good afternoon, all of you.
Good afternoon, sir.
Yes. There are different participants from different institutes arrived here. Let us start with a keynote from Tirkural. Arivatram Kakum Karuvi, Seruvarkum Ul Arikal Aga Aran. That means the wisdom what we gain is a shield for us, even for the enemies who come with the weapons. The wisdom is a shield for them too. So அறிவு என்பது நம்மை காப்பது மட்டுமல்ல, நம்மை தாக்க வரும் நம்மை அழிக்க வரும் சக்திகளையும் காக்கக்கூடிய வல்லதுதான் அறிவு என்பது. So for that in search of that, we are in this time of pandemic also we are moving to gain the knowledge. So the theme which says that in terms of pandemic, advancing scientific knowledge in times of pandemic, we are gathered here. Welcome you all. And now it's my privilege, pleasure to invite our beloved principal madam, to Professor Dr. Grace Ratnam, to gather the welcome speech.
A very good afternoon to one and all. We are gathered here yet again for the 5th in our series of webinar. So I take this opportunity on behalf of the management and staff and our students to welcome each and every one of you who have gathered here. We are in time which the world has not seen for a century, when we had the first Spanish flu epidemic in about 100 years ago. But it is also a time where the world has not seen this kind of technology, which thankfully we are using, and I hope that each and every one of you who have gathered here will be benefited. For today's series of our webinar, we have two distinguished speakers, Dr. Morelli Kumaraswamy. I take this opportunity to welcome you, Morelli. He is not new to us. I have so much pleasure to see him again, because we have seen him as a young, enthusiastic, very talented faculty at our college many years ago. And he has reached great heights today, and it is a joy to see him here, and greater joy to see that he's still connected to these roots, and he has readily accepted, in spite of his busy schedule, to be with us and address our students. And it gives me great pleasure. Murli, welcome. We have with us Dr. Mahendra Shekhar from the University of Kuala Lumpur, Malaysia, and he will talk to us on the natural products So I take this opportunity to welcome you, Dr. Mahendran. Thank you so much for being with us. Welcome.
Now I call upon our Professor, Mr. Balasubramanian sir, to address the gathering with a special note. Sir,
So before that, could you please allow me to share the screen?
yes sir, we'll do.
Yeah.
Vidla sir.
Yeah.
We have with us Dr. Mahendra Shekhar from the University of Kuala Lumpur, Malaysia, and he will talk to us on the natural products. So I take this opportunity to welcome you, Dr. Mahendran.
Hello.
Hello. Hello. Hello, sir.
Hello, sir.
Hello. Hello.
கேக்குது சார் பேசுங்க சார்.
Is it audible? Okay.
சார்.
Okay.
Yeah, sir. Start, sir.
Respected Principal Dr. Grace Natram, respected guest speakers, dear participants of the webinar, good afternoon. This is in the fifth in the series of webinars organized by CL Paidmeta College of Pharmacy, Chennai. It would not have been possible without the active participation by the teachers and the students of pharmacy colleges of Tamil Nadu and the neighboring states, for which we are grateful to them. We promise the present webinar will also be informative, educative, and useful to you all like our previous webinars. Today, we have two eminent speakers, one from the western part of the globe, Dr. Morelli Kumaraswamy of Portugal, who will be giving a lecture on nanomedicine. And we are having another guest speaker from the eastern part of the world, Dr. Mahendran Shekhar of Malaysia. He will speak on the topic, Journey from Basic Research to Innovation in Natural Products. an apt topic at the time of worldwide search for medicine for corona from among all the sources, including natural sources. Many researchers of natural products stop their research at basic level due to various reasons like lack of time, lack of resources, lack of facilities, above all, lack of support and encouragement. Well, this particular webinar will focus some light on the dark road and lead the interested people to the destination of innovation, we hope. With these few words, I welcome the speakers to today's webinar. Thank you.
Thank you, sir. Now I request Mr. Santini Nachiyar, madam, to introduce the speaker briefly.
Thank you, sir. Thank you, ma'am, for your valuable speech. It's an indeed pleasure to introduce our today's speaker, Dr. Morelli Kumaraswamy, Senior Researcher, Israel Institute of Technology, Israel. Dr. Morelli Kumaraswamy received his M.Pharmacy in Pharmaceutical Chemistry from our C.L. Mehta College of Pharmacy. At 2010, Dr. Morelli received a research grant that allowed him to work with the research group at the Nanobiotechnology and Bioanalysis, University of Rovira, Spain. At 2015, he obtained doctorate in theoretical medicine from Doctoral School of Semmelweis University, Budapest, Hungary. 2017, he joined at Mediterranean Technion through the National Network of Excellence in Neuroscience, grant from Israel, largest Teva Pharmaceuticals, and supported by planning and budgeting fellowship from Israel Council of Higher Education. He is an author of over 30 publications and inventor of patent applications. In 2020, Dr. Morelli received European Union Interreg grant to explore his transgalactic approaches on diagnostic and treatment of pediatric brain tumor. He has also received Innovation Prize from Startup Portugal for his innovations treatment approach on COVID-19. We are pleased to welcome you, sir. Thank you, sir.
Thank you. Thank you so much.
Thank you. Welcome, Dr. Morelli Kumarasamy. Now the screen is yours.
Thank you so much. I think because I'm switching off my camera, so
okay.
Because I'm presenting from my home, so my internet is choppy maybe. So okay. Thank you so much. Thank you, everyone. First of all, let me thank the ma'am for the brief introduction. Also, let me thank Vijay Kumar sir, also Prashad Prabhu sir. Good morning, everyone. I'm so privileged to give this virtual talk at the C.L. Mehta College of Pharmacy because, as Principal Ma'am mentioned, I'm the alumni of this college. I studied and I worked as a faculty, so I done a dual role. So in this context, I'd like to thank all my teachers, beloved teachers from C.L. Mehta College of Pharmacy. I remember Professor Ramesh Kumar sir, Professor Shanta ma'am, Professor Nalini ma'am, Ramalakshmi ma'am, and of course teachers from other faculties, Muralidharan sir, Kumaraveli sir, also my friends Vijay Kumar sir, Prabhu sir, Ubaidullah sir, and of course Vijay Gita ma'am, and other few faculties, those who left from C.L. Mehta College of Pharmacy, and also our principal Grace Ratra ma'am. And of course, all non-teaching staffs. I still remember Srinivasa Raghavan sir, also and also other people, the other non-teaching staffs. I'm extremely sorry if I missed any of your name. Of course, Bagudurji. So as I mentioned, I experienced C.L. Mehta College of Pharmacy as a student as well as a faculty. So I feel like today is rejuvenating my old memories in the year 2006-10. Almost 4 to 5 years I spent in C.L. Mehta College of Pharmacy. So I'll start my presentation. I'd like to talk about something. Okay. I'd like to start my presentation today is Nano in the Brain towards Precision Nano Neuromedicine. So this is a snapshot of my presentation. As Principal Ma'am said, we live in a most exciting period of science. We have a lot of technology. For example, 100 years back, when we were affected by Spanish flu, we don't have such technologies. But now, within 62 days, Moderna, US-based pharmaceutical biotechnology, they developed a MAV vaccine. And we have a lot of technological advances at the moment, actually. Today, I'm going to talk about the precision, personalized, individualized medicine. So I'll explain later what is personalized medicine. And also, is it different from usual medicine? My answer is always yes, because it's usually different from conventional medicine. And where it goes, challenges, now how nano comes to neuro. So that's why, I mentioned like nano neuromedicine. And also I'm going to discuss some of the basics about neurological disorders, biological barriers, and motivation of this kind of research. And another thing, the main goals of precision medicine is like, minimally or non-invasive approaches in drug delivery as well as disease diagnosis, and also nanotoxicology. So these are the snapshot of today's presentation. As I mentioned, nanoscale. So I hope you came across this term nanoscale, nanoparticles, nanomedicines, and etc. So in this case, nano means like it's a 1 billionth in meters. So also, in this case, our body is composed of many nanomaterials. For example, atoms like it's like a 0.2 nanometer size, and proteins, DNA bases, viruses, ribosomes, genes, everything comes into this is a nanosized. And also our cells, like animal cells, also is around 10 microns, 8 to 10 microns. So body is completely full of like composites of nano. Actually, that's why one of the famous drug delivery scientists, Professor Kazunari Katako, he mentioned like his body is full of nano hospital. Also, in case if you want more layman explanation, for example, human hair is like approximately 100,000 nanometers. So this is the basic of nano. And using this bottom-up and top-down approach, you can manipulate this nano fabrications. There are a lot of techniques that are available to fabricate nano. And next, if I'm talking about nanomedicine, actually, so what is nanomedicine? Nanomedicine is defined as an application. Okay, yeah, I think I can, yeah, sorry. So nanomedicine is defined as applications of nanotechnology to achieve breakthroughs in healthcare. Also, nanomedicine, not only for a healthcare like drug delivery point of view, even nanomedicine plays a very important role in early detection, prevention, and to improve the diagnosis. So these are the main applications of nanotechnology, nanomedicine. And if I want to say in a very layman term, at last is nano in medicine is called nanomedicine. And what is personalized medicine? For example, personal medicine these days is a new mantra evolving in the healthcare. So in traditional approach, it's one size fits all approach. For example, if you have a disease like one drug, it's applicable to all the patients actually, as you can see here. But in case of personalized, the name itself indicating that the treatment strategy is based on patient's unique genetic profile. For example, every person's they have a different clinical, genetic, genomic, environmental information. So this drives this particular concept. So, and in other way, if I say like a therapeutics being tailored to individual patient based on the predicted response of disease, because some patients in the conventional treatment, they will respond, some patients will not respond actually. So in the personalized medicine, we identify based on the Human Genome Project. So we are identifying the exact treatment options and diagnostic options. That's why it's called personalized. Otherwise, you can say like individualized, more individualized medicines. And what is the major goals of precision medicine actually? For example, there are many goals of precision medicine. The one primary importance is like minimally non-invasive. That's what I'm going to talk in my presentation, most of the presentation. So minimally non-invasive, like previous approaches, it was like invasive approaches even in diagnostics and also surgery. But the current one is like a non-invasive, otherwise minimally invasive in both therapy and also diagnosis. Also, the next goal is like a cell type specific. It's not like off-target accumulation. Like these days, like a more target-based, like a cell-specific based target actually. Also, low cost. Low cost is much more important, especially India-like countries. We need the low-cost approaches. Also, the target-specific, like precision oncomedicine is another booming area because precision medicine plays a very important role in cancer medicine also. And treatment without side effects. That's a very important goals of precision medicine. Also, apart from this, the early detection, because early detection is much more important to treat the diseases actually. Even in coronavirus case, the same, right? So also the next, I'm going to talk about the safety because nano is not only for the application point of view, even nanotoxicology is another biggest problem actually in case of if you want to develop a precision medicine. So these are the main goals of precision medicine. So next, why nano actually? So for example, as a pharmacist, the biopharmaceutical classification system. Its classification system is an experimental model that measures the permeability and solubility of the drugs under prescribed conditions. So it has been divided into 4 categories, as you know, you may come across in your textbooks actually. So based on the solubility and the permeability in vitro, but the sad truth is like the category, the classification system 2 and 4, the sad truth, more than 50% of approved drugs and 70% of the recent drugs, it falls on this particular category. So the main problem is, which means that it suffers from the low solubility and high permeability. For example, if you want to treat many diseases, it should cross the biological barriers. So if you want to cross the barrier, the drug should have like a good permeability actually. So it should be in a good soluble forms. That's why researchers make a lot of efforts to improve the solubility and the stability of drugs, where nanotechnology plays an important role. So how this micro nanotechnology overcome biopharmaceutical drawbacks actually? For example, the challenges in pharmaceutical research, not only drug delivery, even for drug discovery, the big challenges are like off-target accumulation. Particularly, you can consider like a cancer nanomedicine. The existing drugs are like off-target accumulation, so we don't have like more specific targeted oriented. And drug resistance is another big problem actually. I think I'm emphasizing all the scientific community should start work on the drug resistance, because next pandemic is going to come from the drug resistance, antibiotic resistance, and multi-drug resistance. So we need to be prepared for this particular pandemic. And poor bioavailability, that's another problem, as you know. And physical instability, actually, that's most of the conventional drugs is not stable inside our body, because your body is full of a different pH, different ionic strength actually. So these are the main challenges we are facing in drug delivery and drug discovery. And the drug delivery carriers, there are different categories of carriers actually. First, I can say like based on surface charge, like a positive or negative, like cationic, anionic based drugs. And also the compositions, different compositions available. For example, polymers, metal nanoparticles. Also, viruses like non-virulent viruses are used as a drug delivery carriers. Also, the silica, liposomes is one of the successful story comes from the nano and polymeric nanoparticles and extra. These are the main compositions of the drug delivery carriers. In other cases, different geometries are actually also playing an important role. For example, the sphere, rods, triangles, these are the different geometries is used as the drug delivery carriers. And other things is like targeting ligands. For example, we are surface functionalizing antibodies, proteins, peptides based on the receptor target, and nucleic acid, aptamers, synthetic DNA, and small molecules, and most of the polymers actually we are using as ligands. But the problem is, there are a lot of nano drug carriers, but the problem is nanotoxicology. That's a big question mark actually, because how nano-sized materials behave inside our body is a big question mark actually. That's why we are also exploring nanotoxicology in different aspects. And polymeric nanoparticles, I'm working past many years. I'm working in the area of polymeric nanoparticles because of many reasons actually. For example, this polymer is such a wonderful invention discovery. Made by Robert Langer actually, because 30-40 years back, using these polymers, we can able to encapsulate any pharmaceutical materials, large molecules or small molecules, within the polymers matrix actually. Also, there are plenty of advantages of drug delivery carriers. For example, you can say like simple production process, and preparation is mass production is possible, and physical stability of biological traits during storage. These are all drawbacks in the conventional drugs, but using a polymeric, you can overcome all these drawbacks. And also improved stability in encapsulated drugs against enzymatic degradation. That's another advantage, and possibility of sustained control release. And another important thing is because of nano, you can make this polymers into nano, you can reduce into nano. So high intracellular uptake, that's very important actually. And another thing is you can make it possibility of surface modification with targeting agent. As you can see here, you can modify, you can functionalize with antibodies, ligands, peptides, and cationic molecules, so that the surface targeting is specific target is much more easy actually. These are the advantage of polymeric nanoparticles. So far, I discussed about precision medicine, nanomedicine, and etc. And now I'm going to talk about the neurological disorders, biological barriers, and motivation of this particular research. Actually, our body is particularly like there are 100 billions of neurons and 100 trillions of this connections, actually neuronal connections. So in this impossible jungle I'm saying you can easily understand because it's really like a jungle, because it's compact net of neurons. So still we are, as a scientist, neuroscientists and many pharmaceutical scientists, they are struggling to unmap this brain, the tiny organ brain. So what happens because in United States of America in the year 2013 they took a brain initiative to unexplore the complicated tiny organs actually. So afterwards, there are a lot of works they started using nano tools for the neuroscience and to map the brain, to map the single neurons using a nanotechnology. A lot of imaging techniques, a lot of drug delivery approaches, therapeutic approaches has boomed after 2013 actually. So, and if I talk about disorders of nervous system, we know very few nervous system disorders actually, but there are 600 neurological disorders. For example, vascular disorders actually, one of the main problem is stroke, ischemic stroke actually. It's because of high blood pressure and cardiac disease, it leads to stroke. And also hemorrhage, that's another biggest problem actually. These are vascular disorders, and also structural disorders, like for example, brain or spinal cord injuries due to accident or something else, and also tumors actually. Tumors, particularly not only adult tumors, the kids, the small kids, they are getting a tumor, brain tumor, pediatric brain tumor. That's really devastating actually. And also functional disorders, for example, migraine, epilepsy, dizziness, neuralgia, etc., etc. And neurodegeneration, probably everyone is exploring this Alzheimer's diseases, Parkinson's disease, multiple sclerosis, amyloid sclerosis, Huntington's. These are all motor neurons disorders actually. So there are plenty of diseases, but I listed very few here. The problem is, in order to treat most of the diseases, your drugs or small molecules or large molecules should cross this brain's own security system, is called blood-brain barrier. The problem is more than 98% of small molecules and almost 100% of large molecules is not crossing the blood-brain barrier. That's the biggest disadvantage, and it's the biggest mystery behind this actually. That's why Brain Initiative has started to explore this mystery. As I mentioned, brain has two different barriers. One is like an epithelial barrier, which is called choroid plexus. You may come across in neuropharmacology. And also there is another barrier called endothelial. I'll discuss in the later slide. For example, choroid plexus. Why I mentioned solute barriers, I hope you came across this cerebrospinal fluid. It's shortly called as CSF actually. CSF is useful in both aspects. One is the diagnostic aspects and also therapeutic aspects. If you see cerebrospinal fluid is shortly called as CSF, it formed at the rate of 0.5 ml per minute. Okay, it's available totally 200 ml inside our brain. And which 30 ml is present in this lateral ventricle actually. The rest is present in the subarachnoid space. And another unique feature of this cerebrospinal fluid is like every 4 hours once it has been replaced actually. So when you want to develop a drug, you should consider all these factors. Drug should be more stable in this particular cerebrospinal fluid and etc. etc. And based on this category, I can differentiate into 3 different barrier inside in the brain. For example, blood liquor actually, that's a choroid plexus is involved in this. And blood-brain barrier, hopefully everyone has came across this blood-brain barrier. Like it consists of endothelial cells, astrocytes, and pericytes, and liquor tissue, ependymal cells actually. So these are the 3 barriers. If you want to develop a drug should cross all these barriers actually. And hopefully you came across the blood-brain barrier. The blood-brain barrier consists of 3 different cell types. One is like endothelial cells and astrocytes and pericytes actually. these three cells, apart from these three cells, the basal membrane, like extracellular matrix also, this is a big, it acts as a gate actually for the many therapeutic molecules, and also many harmful materials actually. But apart from that, brain also protected by microglia. This is like a macrophages, same like it's present in the nose or any skin. This macrophage is present everywhere, but this brain macrophage is called microglia. It's more unique in nature actually. Also, you know about neurons actually. So these, mostly in my presentation, I'm going to talk about the microglia and neurons. And as I said, it's a mystery behind this actually, right? So still we are exploring this, how it's regulating the permeability, because it restricts undesired molecules. It allows some desired molecules like nutritious actually. So this regulated permeability with unknown details. For example, if I give a layman term, glucose is crossing the blood-brain barrier, right? So same like glucose molecules, it's not crossing actually. So it's the biggest mystery behind this. So what sort of particles or drugs to be investigated and it's crossing the blood-brain barrier? And on these two barriers I discussed, solute barriers and the blood-brain barrier, it passes the undesired molecules to the brain, right? So however, several essential molecules, as I mentioned, and nutrients have somehow entered the brain, so it gives a normal functions, right? So there is a specific mechanism actually. There are 5 to 6 different categories of mechanism behind this. The primary the mechanism is like a diffusion-based. Diffusion-based is the smaller molecules like hydrophobic molecules. It's just diffusing into the cells actually. The next one is like a paracellular transport. Paracellular transport is between the cell junctions. As I said, it's like a tight junctions actually. So this paracellular transport, it's happening is between the cell types, as you can see here. And transport proteins like a carrier-mediated transport proteins. For example, glucose-mediated amino acids and nucleosides, a lot of transport-mediated proteins are. And receptor-mediated, this is a very successful story in nanotechnology because a lot of receptors has been functionalized on the nanoparticles, nanovehicles. They're exploring different aspects of a low-density lipoprotein transferrin receptors. There are a lot of receptor-based approaches actually. And also as adsorptive transcytosis actually, this presumably is relying on transport of positively charged drug molecules, as you can see in the pictures actually. So this is the problem is a non-specific manner actually. In this also, nano is facing a lot of obstacles actually. For example, I used to give a term called corona actually, protein corona, which means that nanoparticles bounce on the surface of proteins actually, because nanoparticle has the active surfaces, so it can bind with any biomolecules. So it's called corona actually. It's not coronavirus. I'm talking about the interactions, protein nanoparticle interaction. So when you're injecting into the body, your blood has like more than 6,000 proteins actually. So any of the proteins can bound on the surface of nanoparticles, and it makes like a non-specific adsorption. And efflux transporters actually, these are ABC transporters, as you know, the proton pump efflux transporters. This is the main reason for the drug resistance actually. So we need to explore this aspect. So these are the main mechanisms of the blood-brain barrier across to target the brain. The next approach, so far I discussed about the basics about neurological and nanomedicine, precision nanomedicine. And now I'm going to talk about this very important area of non or minimally invasive approaches in drug delivery actually. For example, in CNS delivery approaches, as you know, there are different conventional approaches, like for example, invasive approaches. Invasive approaches like, you need, there are different invasive approaches. One is like intracerebral implants, just open up the skull and keeping the drug actually, or drug wafer or something. But also transient BBB disruption using certain chemicals like oleic acid and certain chemicals we are using to disrupt the BBB, the blood-brain barrier to target brain. And intraventricular infusion, as we discussed, directly administered in the cerebrospinal fluid. But the problem is in this invasive, it's you need to somehow open up the skull, and it's completely invasive, and you need a specialized person like a neurosurgeon. That's why these days a non-invasive method has reached a very good approach. It's considered as a good approach actually. There are a lot of methods are available, but particular methods like using colloidal drug nanocarriers. using collider drug nanocarriers, one of the main important things is like intranasally to be considered like a non-invasive approach. In this intranasal, you can directly transfer the any nanoparticles or drug materials by intranasal route to the brain via olfactory system, and also there is another called trigeminal system. Actually, there's two nervous systems. As you can see, directly nose to brain actually do not come across many barriers. So what are the advantages? As you know, as a pharmaceutical scientist, you know about the advantage of nose to brain delivery. Actually, for example, is one important thing is a non-invasive method. And always I emphasize is a painless method and avoids hepatic first-pass metabolism. That's a good. And also, as I mentioned, the blood-brain barrier is a big obstacle actually. But in this route, it can overcome this blood-brain barrier. And another important actually things, the more efficient nanoparticles in the range 100 to 300 nanometers for this particular route actually to target this. So the intranasal delivery emerged as a new approach to circumvent the brain. To deliver the drug into the central nervous system. So this is the approach actually. And in the year 2004, Professor Richard Axel and Professor Linda Bach, they have been awarded for the Nobel Prize in Physiology and Medicine actually to unravel the mechanism behind this olfactory system actually. Before that olfactory system, it was not explored much. They answered a lot of unanswered questions actually. For example, how the brain represents the external world actually. For example, brain is such a delicate system, as I mentioned before. It doesn't have any connection with the external world. But only connection with nose. Nose is the only connection. The olfactory system is the only connection for the brain to communicate with the external system. And this is not like a big science. It's a core of philosophy and psychology and neuroscience actually. A lot of things. So they discovered. They revealed a lot of information in the research. And why is olfaction is important? Why is smell is important actually? For example, vision is important, right? So the same way, the olfaction also is very, very important actually. And also, how does our sensory system work? How does it allow us to interact with the outside world? Actually, these are the very important questions. They answer has been answered actually from the research. After 2004, a lot of researchers they started exploring this olfactory system actually. As I mentioned before, the olfactory system, the central nervous system, and olfactory region is like a tightly packed system actually. Almost there is no extracellular space. Why? Because the olfactory system nose to brain, it connects with the many types of cells. For example, mitral cell, cribriform plate, olfactory receptor neurons, and also behind this epithelial cells, mucus actually. That's a big target for all the pharmaceutical scientists. And there are many cells behind this. And as you know, neurons always is like a billion trillion connections. And neurons are not always alone. It's always combined with glial cell connections, actually. So it's like a dynamically changing system, and it's a delicate system of connections. And there's another unique feature of olfactory epithelium, neuroepithelium, as an extraordinary unique characteristic of the turnover throughout the lifetime. Actually, recently, last year, one of the papers has been published in Nature Medicine. They were talking about even at the age of 95, your brain is giving a neurogenesis, like a neurons, like new neurons are generating actually. So this is a unique nature of our nervous system. And because in another problem with the nurse brain or the brain modeling, the in vitro actually. For example, many previous researchers they were working on animal experiments actually. For example, I'm not like against animal experiments, but the problem is, when you're going for animal experiments, you cannot identify the exact mechanism actually in this jungle, right? As I mentioned, like a jungle is a complicated system actually. So just introducing a drug into the brain using an in vivo model, you cannot understand many mechanisms actually. That's why in vitro models, actually, particularly using exact cell models, is much more important actually. The previous existing models, they use cerebrospinal fluid or nasal epithelial tissues, immortalized nasal epithelial tissues, and nasal epithelial mucus models in vitro. But the problem is, these models are like somehow immortalized, and you do not know exactly what's happening exactly in the in vivo experiment. So in order to overcome this, we use like a primary cultures, which means that we isolated cells from the animals, and we used actually with the different fluorescent nanoparticles. Another problem with the existing models is like a highly simplified, as you can see, it's like a transfer flight models, highly simplified models, and 2-compartment culture dishes with several limitations. And another problem is, as I mentioned, is like deprived of actual olfactory cells actually. So that's why that's why. So you need exact cell compositions to understand the mechanism behind this all. So what we did actually, to investigate the different nanoparticles or drugs actually. So we initially isolated the key cells from the different parts of for the animals, so the olfactory epithelial, olfactory bulb, and also cortex, brain regions, and we thoroughly characterized. I'm not going to discuss very deep in this because due to time restrictions. So we thoroughly characterized this different cell types using a different biomarkers, and we used different nanoparticles actually. For example, as I mentioned, I'm sticking with the polymeric nanoparticles. So we used 5 different nanoparticles in the size range of 17 to 500 nanometers. Mostly we use a chitosan, polymethyl metabolite, different compositions, PVA, and also we used a hydrolyzed galactomannan, which is mostly like it will be considered like a grass polymers, and PEG PCl is another combination, and also we use tetronics and pluronics actually. This PO combinations. We used 5 different, also different sizes and different charges actually. We consider in this, and as I mentioned, we utilize for the actual cells, key cells, and to the best of our knowledge, no previous study actually utilized the purified olfactory neurons to assess the nanoparticle internalization studies. Actually, this is the first study we used. We published this papers in last year in the journal Advanced Biosystems. As you can see here, we exposed the polymeric nanoparticles, as I mentioned, 5 different polymeric nanoparticles with the different compositions, engineered polymers, and our results revealed that the beta tubulin, actually beta-3 tubulin, is specific for the neuronal markers. And if you can see, the immunopositive tubulin is not uptake the nanoparticles. Conversely, the uptake of smaller nanoparticles, here the nanoparticle, you consider that the green color is the nanoparticles. Like we always label the nanoparticles with the green or red. So this green color, so some of the tubulin immunonegative cells, it consumes a huge amounts of particles actually. So there's a possible this is suggesting the possible even in cortex, the same thing, like tubulin positive is not uptaken any nanoparticles. Instead, tubulin negative is uptake the nanoparticles. So it's suggesting the possible involvement of other cell types in the transport system. And afterwards, what we did actually, we isolated the primary brain cultures containing neurons, astrocytes, and microglia. As I discussed in my schematic presentation, this is like a composition of cells actually. So we isolated the primary cultures containing astrocytes, neurons, and microglia. As you can see, when we expose to the nanoparticles, the green color, as you can see, only cells with the microglia morphology, the glia morphology, it accumulated huge amount of particles, whereas neurons and other astrocytes did not take up any particles actually. We also did other confocal microscopic investigations, and as you can see, particles are inside the cellular vesicles. Also, we always used to do 37 degrees centigrade because which is a physiological temperature. Also 4 degrees centigrade because it suppresses all the transporters actually inside cells in order to understand whether this active or passive approaches, active or passive mechanism. In the 4 degree, as you can see, the particles are just accumulated on the surface. So as I mentioned, neurons did not involve. Some type of glia is involved in this. So we purify the astroglia And again, we expose polymeric nanoparticles, the green color polymeric nanoparticles. In this case, what happens, particularly not all astrocytes, but only some selected astrocytes taken up these particles. And so also we repeated the same experiments with the plus 4 degrees centigrade. As you can see, in the plus 4 degrees centigrade, the particles are just on the surface, which means that this is like an active uptake. So many transporters involved in this. So only some selected astrocytes. So as I mentioned, there's a possible involvement of other cell types, right? So here, somehow it's struck actually. Okay. So what we did afterwards, we isolated microglia from the mixed culture using a defined media, as you can see here. Can you able to see the video? Yeah. We isolated a microglia, actually, one of the macrophages present in the CNS, as you can see here, using a defined media. So other cells has been separated, or other cells means here the neurons and astrocytes has been removed off, and microglia stays back. And in this case, if you can see microglia, how this microglia is achieving other debris or any cellular materials, as you can see in this picture. Can you see the microglia, how active, how it achieve up any debris actually here. Also in this case, you can see this is a debris, cell debris, the microglia is eating actually. So why I'm showing this video, the same way when any pathogenic materials, organisms enters into your body, for example, virus towards nose, the same, our macrophages doing the same mechanisms. That's why the cytokine storm, a lot of cytokines and inflammatory cytokines is secreting actually. So this is the thing. Then after separations of microglia, so we exposed we exposed the same polymeric nanoparticles into the olfactory and cortical microglia. And in a 1 hour exposure, as you can see, microglia cells accumulated huge amounts of polymeric nanoparticles actually. In the previous case, what happened? Neurons did not take up any of the particles, but in this case, the microglia, like macrophages, which is present in the brain and also in the olfactory regions, it accumulated significant amounts of polymeric nanoparticles. So this indicates the possible involvement of the microglia, like the phagocytic cells in the nose to brain transport. Actually, also this particular cell is also expressing the INS, like nitric oxide synthase, which is one of the stress marker. This particular marker expressing cells also accumulated huge amount of nanoparticles on. Also, we performed the experiments with the highly purified microglia cells obtained from the transgenic animals, the fructose receptor modified transgenic animals. As you can see here, you consider like a red is like nanoparticle. The microglia accumulated this Nile red polymeric nanoparticles. And the next, what we did, we exposed different nanoparticles, but I'm showing only one nanoparticle here as an example. We exposed hydrolyzed galactose nanoparticles, the polymeric nanoparticles, onto this microglia. What happens is the olfactory microglia, it forms like a lipid droplets actually. So this lipid droplets is really unusual actually for me, because when I first experienced this, I was really doubting about this discovery, because the lipid droplet particularly it's very important mechanisms for the adipose tissues, as you know, like fat tissues, adipose tissues, and another important things for the cancer cells particularly, because cancer cells it secretes lipid droplets, as you can see, the small dot kind of thing. Cancer cells mostly it secretes the lipid droplets as a against mechanism, as a defense mechanism. So when we further analyze, this is a schematic of the lipid droplets. When we further analyze with the ultrastructural analysis, as you can see, the electron microscopic images shows like a lipid droplet with white big ball-shaped things is like considered as like a lipid droplet. Then we identified its lipid droplet accumulating microglia. So this lipid droplet can act as a early and robust marker for the immune response initiation. Actually, even any external materials goes and attack our immune system. This kind of lipid droplets is like a marker, biomarker for this for the immune response initiation. So also this main indication of reactive oxygen species, hopefully you came across, and also pro-inflammatory cytokines. So then what I did actually, so this is a dancing video I like to show you. See how the microglia, isolated microglia, is active actually. You see how it eat up this debris actually, which is present inside the cell culture. It's in vitro videos, but still the microglia is like really active and eat up all this debris, actually unwanted things. And next video, how it's active actually. It's a never-resting microglia. Always is monitoring our brain environment. Any harmful materials if it enters your brain, this microglia will be activated, and it gives like alarm sign. And also. Further, we analyzed our structures using transmission electron microscopy. It shows as microglia contact specialized areas of neuronal bodies. As you can see, this is a microglia part and also a neuronal part actually. So the same way as I shown in the schematics, the microglia always is monitoring the neurons. So we identified microglia is protecting the neurons in a certain specialized areas actually. Recently, that particular area was not explored. Recently, one of our collaborative group, they found there is some purinergic junction is between this microglia and neurons. So this is why microglia act as like a protection for the brain environment. Also, we did some experiments with the self-assembled mini brain, as you can see in the video. We exposed this green color indicates the polymeric nanoparticles. We exposed the polymeric nanoparticles into this mini brain containing all 5 different cell types like endothelial, astrocytes, pericytes, neurons, and microglia. As you can see, this the green color, particularly it was certain amount of cells actually. So which indicates this microglia is monitored and protects brain function. When you are exposing the polymeric nanoparticle, the microglia come and capture this particular nanoparticles or any harmful materials. And based on this, our research actually, so I'm summarizing my research. Inherent defense mechanisms, olfactory region, particularly the nose to brain region. The mild injury, astrocytes, the astroglia I mentioned, becomes phagocytes and remove foreign materials, produce anti-inflammatory cytokines. And in case of X injury, reactive astrocytes produce pro-inflammatory cytokines and activation of microglia, CNS resident macrophages. The similar mechanisms will be applicable for any invading pathogens. For example, virus pathogens, the similar mechanism is applicable. So our findings reveal that neither olfactory nor forebrain neurons take up the particles. Conversely, the olfactory and cortical microglia phagocytose the particles, the polymeric particles, independently of the features actually. So neurons are not, the microglia is yes. So why I emphasize this point, the previous research, most of it is more than 100 papers they published actually. Everyone they claim based on the in vivo experiments, they claim the olfactory neurons are responsible. But ours is the first investigation. We claim that olfactory sensory neurons is not responsible for this nose to brain transport. And only the microglia like this macrophages involved in this nose to brain transport. We also done some non-invasive in vivo imaging, as is mentioned, the precision medicine goals. One of the main goals of precision medicine is like non-invasive or minimally invasive. So our approach is not to sacrifice any animals actually. So we did intravenous and also international approaches using a different polymeric nanoparticles in the different time exposures. And this is one of the IV spectrum. This is one of the non-invasive imaging facility we utilize, and we found particles inside the brain actually. Even after intravenous, we found some particles in the brain and the spleen actually. I'm not going to discuss those details actually, which was not published yet. Also, I mentioned in the mechanisms, right? There are different mechanisms. One mechanism is like a receptor-based mechanism. One of our investigation, we functionalize the polystyrene nanoparticles, functionalize the cyclic RGD, which is like more specific for the integrin receptors. When we expose into the primary cultures containing neurons, particularly, as you can see, nice structures of the neurons, this particular cells accumulated huge amount of the surface functionalized, peptide functionalized particles. So which indicates is like a receptor based actually, receptor based interactions. And so far I discussed about one of the methods of drug delivery. I'm going to discuss another approaches in drug delivery, non-invasive approaches actually. The nose to groin and the another minimally invasive or non-invasive approaches is to tackle this blood-brain barrier is called focus ultrasound mediated drug delivery to the groin actually. This is one of the safest method has been developed in the past 3-4 years. A lot of clinical trials is going on this particular approaches actually. As you can see in the picture, you can apply a focus ultrasound after exposing a drug molecules. So what happens is the blood-brain barrier is the biggest barrier, as you mentioned, but using the focus ultrasound, this endothelial cells is one of the tight junction cells present in the brain. It opens up the tight junction so that your drug molecules can pass into the brain. So this way you can improve the dosage, and it's the best method for many neurological disorders actually. The advantage of this method is a non-invasive methods, is no surgery needed, and precision targeting, that's very important, and treatment to affected areas, only affected areas, no like off-target accumulation. It's minimized damage to the brain, and another important factor is like open BBB, the blood-brain barrier safely, and enhanced drug delivery and immune effects actually. And we also started some collaborative works with the University of Pittsburgh Medical Center. So we are going to work on like Alzheimer's disease-based aspects actually this time. And not only that, we also performed some experiments with the focus ultrasound, but not on brain tumor aspects, but on rhabdomyosarcoma. So one of the a devastating disease is a cancer, very early forms of muscle cell cancer. It's more common in children, but sometimes occurs in children also. I mean, adults also. It happens in the muscles, as you can see here. So what we did in this case, we did only in vitro experiments, not like in vivo. We passed ultrasound on this human rhabdomyosarcoma cells, as you can see in the pictures, and we analyzed different effects actually. Our results shows when we expose our polymer ceramic nanoparticles and when we apply this ultrasonic focus ultrasound for 10 minutes, what happens, as you can see in the scanning electron microscope pictures, some cells got shrinked actually. It's just, this cell shrinkage is the main indicator of apoptosis. And also we further analyzed using mitochondrial approaches actually. As you can see, this red color indicates this dye go and bounce on the mitochondria. So which indicates the mitochondria became leaky after these treatments, and so this dye shows inside microglia. And also we did another further investigations on ultrastructural investigations. As you can see in the picture, mitochondria somehow got damaged actually using our approaches. So this is one of the in vitro approaches. In vivo is ongoing in laboratory. So these are so far I discussed about minimally or non-invasive drug delivery approaches. So now I'm going to talk about disease diagnosis. As I mentioned, precision medicine goals is not only delivery healthcare, diagnosis is also very important, right? So then next, I'm going to talk about the non-invasive or minimally invasive approaches. So for example, blood-based biomarker discovery actually. This biomarker discovery is important not only in diagnostic aspect, even important in a therapeutic aspects actually. For example, as I mentioned in the initial presentation, the brain tumors, not only in adult actually, the children brain tumor is really devastating. Can you imagine like 2 years or 3 years old small kids? They are also affected by this brain tumors. So what's this approach, the blood biomarker approach? In this approach, a lot of technology involved, particularly the medical device, lab on a chip, microfluidics. A lot of research is going on. Actually, I'm not going to discuss so many things actually due to time restriction, and also I don't want to deviate you actually at the moment. It's a simple approach. If you have a tumor, in a brain or something, the tumors, it's that's called metastasis. Actually, it spreads all over the body. Particularly, it spreads into the vascular system, vascular circulations. So a lot of research is going on to find early diagnosis, early detection using a circulating tumor cells. Actually, so early diagnosis it makes therapeutic easy, and you can find the diagnose, like you can find the cancer at the very early stage, so that the treatment the treatment option is much more easy. Actually, there are a lot of markers. Actually, one of the markers is the circulating tumor cells, and also circulating DNA, the methylation changes, and circulating mRNA, exosomes. There are different approaches. Actually, this is one of the personalized treatment approach. As I mentioned before, every patient is different. We have different stage, different heights, different weight. Actually, so these approaches, you can identify, you can read the each patient history based on your blood profile, and there are a lot of techniques, sequence, high throughput methods are available, RNA sequencing, transcriptomic approaches, and etc. etc. And another approach, if I want to say, the blood-based biomarkers, you can, after analyzing the blood, after collecting the blood, the macromolecules, you need to analyze, right? There are two different analytical techniques, actually. One is like a label basis and non-labeled, actually, I categorize. So label basis, for example, this qPCR, I hope you came across mostly the COVID diagnostics based on the qPCR, actually. Also, there are a lot of ELISA-based mass spectroscopic measures, metabolomics measures, actually. There are a lot of research is going on in this particular area. The problem is this is a bit expensive. You need to, somehow use expensive chemicals. So that's why the non-labeled materials, non-labeled approach, using a spectroscopic approach, mass spectroscopy, LCMS, chromatographic approach. Very importantly, Raman spectroscopy is involved in disease diagnosis. Actually, that's a booming area. Even we can explore this aspects, Raman spectroscopy. And one of our project, actually, recently we got funding from the Interreg, Spain and Portugal. So for the pediatric brain tumors. So the main concept is, for example, using a micro NMR. NMR is not only for the chemical analysis, even you can use it NMR in the diagnostics, actually. Same like Raman spectroscopy. You can use. So in cancer cells, like in cancer stem cells, particularly in brain tumor cancer stem cells, the ferritin is one of the proteins is expressed. The ferritin is name itself indicating it contains some of iron, right? So using this iron, we are detected using this micro NMR approach. As you can see in the picture, you need just one drop of blood, not even one drop. It's just microliters of blood is more than enough to identify this using this micro NMR approach. Actually, also we are applying artificial intelligence. That's one of the booming area, machine learning. So I'm not an expert, but just we have started learning, actually. So after analyze, we are also applying to diagnose the pediatric brain tumors. Also, we are collaborating with the University of Santiago Compostela and also hospitals for the clinical samples. And I'm going to the last part of my talk is nano safety, nanotoxicity. So far, I discussed about the application point of view. So now I'm going to talk about nano safety, actually nanotoxicology. I know many people, many publications. They are utilizing silver nanoparticles. Silver I mentioned because a lot of nanoparticles are toxic. Mostly inorganic nanoparticles are toxic, but silver I mentioned, a lot of commercial products are made up of silver. So for example, men's socks, masks, bandages, sunscreen, cosmetics, and a lot of commercial products obtained they contain silver nanoparticles. The problem is Argeria. So long back, this man, he consumes a huge amount of silver. So what happens? Silver when he consumes silver becomes like ionized, actually, because silver is like inorganic nanoparticle. Inside the body is like a different ionic strength, right? So all the inorganic nanoparticles, it gets ionized. This silver gets binds with the hemoglobin, and it became a bluish man. Its skin color got bluish, actually. This term is called Argeria. So bluish man. Afterwards, this man died. So. And not only in that aspects, long back there was a study, actually. So they performed a silver nanoparticle biodistribution studies using a rat model. What happens? Silver nanoparticle reaches the brain, but the problem is, they confirmed in the brain, but no particles were detected, which means that they detected only silver ions, not silver particles actually. As I mentioned, brain, our body has different strength, different pH level, even cells, some parts like a acidic environment. So in this environment, this particular metal particles gets ionized actually. Okay, gold is like a different story, but silver is like a different story. So we need to consider while using this metal nanoparticle, particularly silver and all. And nanoparticle in the central nervous tissue, actually, I made this picture because, these days we started using a nanomaterials, both accidentally, nanoparticles can go to your inhalation, like nose to brain, nose to lung. There's a possibilities of nanomaterial exposure in different ways, actually. Because of acceleration of both manufacturing skills and intention to use. So this is a big problem of nanomaterials, and routes of invasion, permeation, and distribution of various cells and tissue compartments. That's we need to identify, and effects on different types of neural tissue type cells. Actually, the problem is, as I mentioned, particularly the nanoparticles has active surfaces, so it can bond with any sort of materials like biomolecules. So excretion is the biggest problem in nanomaterials. So that's what nanotechnology is facing the difficulties in translation. And in our own experience, I'd like to share actually, because so far I discussed about nano applications, and also I touched some nanotoxicology. And in our own experience, when you're using a polymeric nanoparticles, particularly using a PEG actually, polyethylene glycol and all. As a pharmaceutical scientist, the endotoxins, like bacterial endotoxins, are ubiquitous contaminants of materials. Actually, not only the materials, not only the parental products. It also the biggest problem for including nanoparticles actually. So in this case, what we did, we measured the potential endotoxin absorption of polystyrene functionalized with carboxylated and pegylated actually. PEG is more safe, right? But in this case, what happens using a LAL-assay? Unfortunately, I have to use this word like unfortunately, we found the particles gave this positive LAL reactions. So, which means that the particles got contaminated with endotoxins, which is the biggest problem when you are using your particles in vivo or certain applications like brain applications. So. With this, I'm finishing my presentation because Dr. Magendran is waiting. I don't want to kill his time, and I thank my group from Israel. I already left Israel, but still I have a very good connection because I have to go back for a while. I came back, but afterwards the pandemic started. I'd like to thank my group from Israel, and also I'd like to thank the Technion. It's called the Mediator and MIT with 3 to 4 Nobel Prize winners. Einstein is involved in Technion, so one of the prestigious institutes. Also, I'd like to thank my prestigious fellowship like Marie Curie Fellowship, one of the prestigious in Europe, and Teva Pharmaceuticals for funding our nose to brain transport studies, and European Commission for different fundings, and also Council for Higher Education, Israel for funding different things, and also my European partners from all part of the European Union. And thank you so much, and stay safe. And I'm looking for a widely collaborative and successful project. And thank you all for your kind attention. Once again, I thank CLB Meta College of Pharmacy Administration and Principal Ma'am, Grace Rathan Ma'am. Also, all my beloved teachers, also my friends in CLB Meta College of Pharmacy. And thank you for your kind attention. And thank you so much. And if you want to email me, you can email. I still consider as a student of CLB Meta College of Pharmacy. So any students, you can feel free to email me, ask for any details or any fellowship, any scholarships. Feel free, and thank you so much. And thanks a lot actually. And yeah.
Thank you, Murali. Thank you. I think a lot of questions are there. I call Ms. Sheila Madam to present the questions with our presenter, Dr. Murali. Sheila Madam.
Sir? Okay.
Yes,
Thanks for sharing your yes, sir. Thanks for sharing your course in subject knowledge, sir. There are some questions from participant side.
Yeah.
The participant named Dr. Selvakumari asked one question. Can you please give clarification on nano carriers from natural origin?
Classification nanocarriers from natural origin, actually. For example, a lot of natural polymers, actually, we are working on. You know, if you categorize the polymers are categorized by natural polymers, synthetic polymers, actually. So a lot of natural polymers, for example, chitin, actually, it comes from the natural products. So and still a lot of marine-based polymers we are characterizing, we are utilizing for the drug delivery applications, not only drug delivery, even for the tissue engineering applications. So a lot of successful stories from natural origin, actually. I hope I answered the question. Yeah.
Okay, next question. With in vitro cell line models, more opt to find blood-brain crossing index of the drug molecule.
Sorry, I'm sorry. I'm extremely sorry. I couldn't hear. Could you please?
Yeah. Okay, sir. With in vitro cell line models, more opt to find blood-brain crossing index of the drug molecule.
Okay, which cell line models, actually? There are different cell line models people are using, actually. I would suggest one human-based model which was available commercially. It's called HCME3D3 something. It's available from the Sigma, actually. It was available from Merck Millipore. That's the only human-based blood-brain barrier. It's available market like commercially. Other blood-brain barriers models are available in like a rodent-based, rats, mice-based, actually. But I will not suggest those models, actually. Use this human-based models. The one is commercially available. It's called. It starts with HCME3 something like this. I don't know. You can search in this way, actually. You can easily find it in the internet.
Okay, sir. Next question. What are the opportunities in nanotechnology?
Opportunities in nanotechnology. Actually, you should ask this question 10 years back. 10 years back, nanotechnology was a really good, big hype. That's the reason I chose my life in nanomedicine, nanotechnology. There are some opportunities, actually, at the moment, even application side. For example, you can consider nanotechnology in nanofabrication aspect, like nanofabrication, like physics-based approaches. Actually, lot of technology-based developments using in nanotechnology. For example, 20 years back, even I still remember when I was in CLB at the College of Pharmacy, I used a flash disk of 256 KB. Like, I'm talking about pen drive. So, but now I'm using terabytes, actually, gigabytes, terabytes, something like this in a very compact. So that's a successful story of microtechnology, nanotechnology. So a lot of opportunities in that aspect, actually. So yeah.
Yeah, last question, sir. You told about micro NMR. The difference is only the sample amount when compared to other NMR techniques. Is this micro NMR technique is available in India?
Actually, micro NMR I was talking about is a very compact system. Just you can carry in your it's like a same like your laptop, even less than your laptop. It is not available in India because this technology, which was developed by MIT professors in the year 2015. You can see it in the publications, actually. My first postdoc using this very basic of magnetic approach, actually. In malaria, when you got infected, the malaria infected bloods contain hemazoin. It's like a magnet. That hemazoin crystals is present in our blood, actually. So that hemazoin crystals has a magnetic properties. So first my research started after my PhD in analyzing the hemazoin crystals using the micro NMR. So India doesn't have this facility because still it's in research. A lot of clinical approaches are going. It's MIT technology, MIT and Singapore technology, so it is not available in India. But if you want, I used to suggest, even in previous seminars also, I suggested, like, pharmacy means, like, don't go narrow with pharmacy. You can think in wide aspects, like cross-disciplinary, multidisciplinary aspects. You can think about applying pharmacy in a technology aspects. You can able to make it like a micro mass spectroscopy. That's an easy one. Raman spectroscopy is available. A lot of technology, you can make it like a, you can think about mass spectroscopy in if you want to discover something actually. So, yeah.
Okay, sir. And one more question. Any example for non-virulent virus or targets?
Example for non-virulent?
Virus.
Okay. Yeah, I actually suddenly it's not clicked in my mind. There are a lot of virus approaches, non-virulent drug delivery approaches. I don't know actually. There are some virus, lot of drug carriers actually, non-virulent carriers are available actually, and even you can search in the publications. I'm extremely sorry, I couldn't remember the exact the name of this particular virus carrier.
Okay, sir. Thank you for your clear explanation, sir.
Thank you, Dr. Morelli. Thank you. You will be the inspiration for young one for here, and it's a good, very elaborate speech given. Thank you very much.
Thank you so much, sir.
Next, we'll go for a next session. Now I call upon Shantini Nachiyar madam to give a brief introduction about our next speaker, Mahendran Shekhar.
It's our honor and pride to introduce our today's speaker, Dr. Mahendran Shekhar, Associate Professor and HOD of Pharmaceutical Chemistry, Faculty of Pharmacy and Health Sciences, University Kuala Lumpur, Royal College of Medical Sciences, Perak, Malaysia. Dr. Mahendran Shekhar received his bachelor degree in pharmacy 2004, master degree in pharmaceutical chemistry in 2007. At 2008 to 2011, he was granted a junior research fellowship from University Grants Commission to pursue his PhD degree at JSS College of Pharmacy, UT. He has received several prestigious awards at the national and international levels of our contributions in academic and research activities. He also received the Outstanding Researcher in Pharmaceutical Chemistry Award, International Achievement Award 2018 from Education Expo TV Company's rating system. His innovative products won the gold medals in the 3 consecutive years, 2017 to 2019, Best Innovation Product Awards. He has more than 50 publications, including ISI and Scopus Index journals, proceedings, guidelines, and book chapter, reviewer of number of scientific journals and international standards. We are privileged to have with us at this session, sir, at this moment. Welcome you, sir.
Can you allow me to share my slide?
One minute, sir. We'll do it.
Yeah, thank you. It's okay.
You can share your screen. Mahendra, you can share your screen now. Mahendra?
Can you see my slide now? Hello?
Sir, yes, it's visible.
Okay.
Make it full slide view. Mahendra, make it slide view. Make it slide view.
Is it okay now?
Yes, okay, yes, super.
Hello.
Very nice.
Okay.
Yes, good.
Yeah, good afternoon to everyone, and thank you very much to Madam Shantini for the kind introduction. And I take this opportunity to thank Silvaid Mehta College of Pharmacy and Dr. Grace Ratnam Madam for the invitation to this webinar presentation. So the topic I was selected for my today's presentation is a journey from basic research to innovation in natural products. So why I was chosen this topic is in pharmacy discipline, actually we do a lot of research and publications, and especially based on natural product, because most of them, those are doing lab-based research, they prefer to do in natural product-based research, because natural products are safe and effective when compared to the synthetic chemicals. So in any countries, the economic development is not only based on the research, it is mainly depending on the innovations. And so in my presentation, I'm going to highlight some how we can get innovative ideas and how we can convert those innovative research into innovative products. So that is what I'm going to highlight in my presentation. Yeah. So this topic, so we are going to discuss more on research and innovation. So we must know what is the difference between research and innovation. So research means it is research, which may be addition or an improvement of an already existing technology. So we are just continuing somebody done research previously. So based on the results, we continue the study. That is called research. So innovation means something new, novel, and original, and which have a great impact in society. And that product can be commercialized and it is useful to the society. So that is called as innovation. And research is a component of innovation and is situated at the front end of the innovation life cycle. So normally we cannot avoid research also because research will lead to innovation. So we need some kind of a background of any kind of innovation. So that we can get it from research. So in pharmacy courses in India and all over the world, we have different kind of courses, especially including we have diploma program, we have bachelor pharmacy, we have PharmD, we have master pharmacy and PhD programs, and we have around 3,000 institutions offering pharmacy course. But the nation's innovation contribution in the world is not significant. So the recent evidence state that India got 60th position in the Global Innovation Index, which was stated in 2017. We have 3,000 institutions in pharmacy, and we do a lot of research. But the innovation side, we are a little bit weak when compared to the other countries. So in my presentation, I'm going to highlight about the natural products. So especially pharmaceutics, pharmacology, pharmacognosy, and pharmaceutical chemistry background discipline, they do more research on natural products. Even other area of research also, which is including pharmaceutical technology and pharmaceutical biotechnology, they do more research on natural product research. And the type of research which including academic research, because normally in our laboratory, normally in India, what we do academic based research. We are not collaborating with some industries. So that is why we cannot able to come up with a product into the market. So what we need to do, instead of doing only academic related research or industry related research, so we need to combine. So we need to have a collaboration with industry. So it is academic and industry related research. So that kind of research only we can bring those products into the market. Otherwise, whatever the research we do in the natural based things, that will not be come into the market. So in part of career opportunities for pharmacy students, so we are producing community pharmacies, we are producing hospital pharmacies, and also we are producing academicians, industrial pharmacists, research and development students. So they can do more research in the industrial side, but we are not producing the entrepreneurs. So entrepreneurs means it can be defined as identifying and creating new opportunities within the certain environments. Especially entrepreneurial outcomes in education refers to new venture development, business startups, and job creation, contributing to economic growth and development. So what we need to do, we need to change our attitude from research to innovation. So the students, if they do more innovation, in future, they will be the job creators. They are not the job searchers. So after completion of their course, at least they have some idea how to make a product, how to go for registration, and how to do commercialization of those products. So that only entrepreneurship will play a major role, especially in the pharmacy side. So in my presentation, I made it into two parts actually. The first part I'm going to cover what are the ideas to develop an innovative mind. And the second part I'm going to highlight how you can convert the research into an innovative products. So I put dozens of ideas, means around 12 ideas to develop an innovative mind to the students. So the first one is we have to maintain a keen sense of things. So the most innovative people in the world are also the most inquisitive among us. So they ask a lot of questions. Means we need to have the people who is having capability of asking many questions. So they'll be having the more innovative mindset. So they can be an innovator. So because they'll be asking questions all the time. So then only their mind will be more creative and they can come up with some innovative ideas. Sometimes the greatest insights come from our interactions with people who hold views opposite from us. So normally we avoid the people who is having the opposite views. So that is not correct actually. So what we need to do, we need to take their ideas, we need to get information from them because they are the one who will be giving the opposite ideas. So whatever you have been thinking, so they'll be giving some kind of opposite views. So that will create more ideas for you. And the second one is, hello, yeah. So the second one is jot down the leads and ideas. So whatever the ideas that we get it, we need to jot down because every day wherever you go, maybe you can get some kind of ideas. You may plan to innovate something, but later you may forget it. So what we need to do immediately once you get an idea, we need to jot down, and you need to make how that can be implemented. So you will need something to capture and organize your ideas and thoughts as you go about your day learning new things. So try jotting down everything that comes to your head, including random words, phrases, and even doodles. And the next third one is the seek of new experience. So to think outside the box. So, hello. Yeah. Normally, we need to think outside the box. So normally, pharmacy students or pharmacy academicians, what they think more on only on the pharmacy side. So what we need to do, we need to think something out of box. Then only we can come up with some kind of innovative products. Means we need to have a collaboration with engineering people and also technology, because then only we can come up with a product. Otherwise, with the pharmacy only we might be having some ideas, but cannot come up with a product. So what we need to do, we need to think outside the box, and you must step out of our comfort zone. So normally pharmacy people, they are comfort with our own research. Whatever the research they do, they are comfortable with that, and they don't come up with something new thing. So those we need to come out from that. Then only we can create new innovative products. So the greatest innovators are often individuals who learn from different fields and merge them together into something extraordinary. So we need to merge with some other program, some other faculties, or maybe the institutions, or something like engineering or technology people, so that we can come up with an extraordinary product. Otherwise, if you do only with the pharmacy, so definitely the product cannot go up to the market level. And the fourth one is we need to practice mindfulness. So it is a continuous attempt to pay attention to what is going around us. And so we need to focus on the present. So whatever the present things that are required. So based on that only we need to make innovative products. So you for example, your innovative ideas or maybe product which may be existing almost 10, 20 years before. So it can make a similar kind of product that cannot be useful. So what we need to do, we need to make a product something which is useful to the current environments, current people. So focusing on only one time is also very important. So because your mindfulness is very, very important. If you focus on many ideas or many things, then definitely you cannot come up with a single product. So what we need to do, you need to focus on one idea, then based on that you create more and more ideas within that same innovation, so that you can come up with the final product. Hello. Some interruption. Yeah. Next one is we need to take risk and make some kind of opportunity. So see mistakes as opportunities for perfecting their innovation. So what we need to do, sometimes we make mistake when you do research or maybe make innovative products. So what we need to do, we need to learn from the mistakes. So mistakes as considered as an opportunities for get a perfect innovative product. And next, you need to share your ideas with others. Something like you will definitely need feedback from your innovations, because sometimes you may think the innovative ideas, whatever you get, it may be something very big. But when you talk to other people, they may think this is nothing actually. So something, that is why you need to have some kind of discussion with other people and sharing your innovation ideas, so that you will get the idea will become stronger and stronger and better. So finally, you can come up with a better product. And the next one is mistakes and failures are inescapable as we experiment. So we cannot escape from the mistakes, and definitely you may face many failures, because bringing one innovative product into the market is not an easy task. So you need to face a lot of negative things also during the experiment. So some people may call it unrealistic idealism. So whatever the ideas that you may get it, if you share with other people, they may tell this unrealistic. This is not possible at all. So, but prolific innovators like Thomson Edison, had to find 10,000 ways that didn't work before succeeding in creating the light bulb. So he tried for almost 10,000 times to come up with the final light bulb. So similarly, so whatever the failures you get it during the innovation product, so you need to come up with that one. You need to overcome those disadvantages and failures, and finally, you need to make your idea to be the realistic one. So you need to try more times. And the next one, preserving with the laser, like focus on your goal is great, but sustaining attention on your task for an extended period of time can defeat your creativity users. So you need some kind of relaxation also, because every time you are thinking about something about innovation, because innovative minds, you need some kind of relaxation also. So take some breaks alone by going for long walk and taking showers and exercise, or indulge in daydreaming to connect with your subconscious mind. So because your mind is, because subconscious mind is very, very important. So if you are thinking too much about some certain innovative products all the time, so definitely you cannot come up with the final solution, because your mind is almost with full of so many ideas. So what you need to do, you need to have some kind of relaxation, so that will be more useful to get a better idea. Then next will be, so you need to build on existing ideas. So some people, very difficult to bring a new ideas. Totally new means, I think it is very difficult for most of us. So what some innovators they do, they don't just invent something out of nothing. So they build on existing ideas. So means what are the ideas that have been created by others? So just they will do some kind of a modification. So something like many products we see today are the result of merging two or more ideas or concepts together. So we can see many mobile phones and cars. So every year they're introducing a new product. How they're introducing a new product? So they come to get ideas from the customers. And what they do is to overcome whatever the disadvantages from the customers. So they come up with a new model or new product. So that way, that also considered as innovation. So innovation is not like something it should be 100% new. So we can modify the existing product with customer satisfaction, that also considered as an innovative product. So a true innovator must open him or herself to as many potential ideas as possible, and identify patterns and connections between them, and finally assimilating them together to come up with an innovation which is better than before. So it is some people, they may tell this kind of product is already existing one, so why you want to come up with a new product similarly like that. So what you need to do, your explanation is supposed to be, so you have to tell what are the disadvantages of those products and why you are interested to come up with this product. So the new product may overcome what are the disadvantages that was faced by the customers. So it is considered still as a new innovative product. And the next one is how you can get an idea. You need to ask the customers. So because customers are very, very important in innovative products, because finally, whatever the innovation you do, whatever the products come to the market, finally it goes to the customers. So what we need to do, we need to take some kind of survey with the customers. So how you could improve your product or service, and they will give you plenty of ideas, because they are the users, because they know what are the disadvantages of your products. So based on that, you can change or modify your product, and that can be as a new product, and you can come up with a cheaper, faster, and easier to use some kind of products. So listen to these requests carefully and choose the ones that will really pay back. So some customers, most of the customers, they give the very good idea. So based on that, you can come up with a new innovative products or ideas. And the next one is use difficulties and complaints. So some they may think if the customers are complaining or customers are telling difficulties, so that they may think that is disadvantages. So what we need to do, we should take that as advantages. So what are the difficulties they are saying? So you need to overcome those disadvantages. So you need to come up with a new product. So if customers have difficulties with any aspects of your product, so if they register complaints, then you have a strong starting point for an innovation. So if you get a complaint from any product from the customers, so that is a strong starting point for your innovations or inventions. So make your product easier to use and eliminate the current inconveniences and introduce improvements that overcome the complaints. So then next one is you have to use social media. Nowadays in social media is playing a major role, because when you put any kind of ideas or if you put any questions in social media, very fast you can get some kind of reply or answers from many people all over the world. So they will give certain feedback. So that feedback will be very useful, and from that way also you can make future products. So many early adopters are active in social medias and social network groups, and they will be happily respond with suggestions. So these are the ways that we can get ideas. And next, I'm going to discuss more about that, because my topic is more related to natural products. So I'm going to discuss how you can, what is the current practice in natural product research, and how we can convert those research into a innovative products. So every one of us knows that natural products includes a large diverse groups, and we know that natural products, I think some interruption because some sound outside. Natural products means it is mainly obtained from plants. It also obtained from bacteria, fungi, and also insects. So discovering promising new candidates from natural products are extremely difficult, expensive, and time-consuming process. And due to this, many scientists focus on developing and testing synthetic molecules. So now, natural world provides huge diversity, and we have so many new products, including the olden days, we come up with so many products like penicillin, and also penicillin from penicillin fungus, and anti-malarial drugs like chloroquines. These are all from the natural products. So that is why the natural products are playing a major role even still nowadays. So very many people they prefer to go for natural products. So our researchers also normally they put a lot of efforts towards the discovery of new natural molecules to fight against diseases. And many institutes and centers research programs that discover, design, develop, and that novel compounds for use as therapies. So normally when we do a natural product research, what we do, we start with the selection of plant material, which including literature review. So based on the literature review, only we select the plant material, and also we also do checking traditional and agro-medicinal uses. So based on this only we do selection of plant material. So after selection of plant material, we go for authentication. So the plant authentication done by certified botanists. And identification is very, very important, and molecular specimen also very, very important to select the plant material and authentication. So basic operation steps, which is including pre-washing and drying the plant material. So normally for drying the plant material, we use pre-dryer. Proper action must be taken to ensure that potential active molecules are not method of during I think there is some interruption from the outside. Normally, the basic operation is including pre-wash. And after that, we go for selection of plant material. So,
audio is not clear.
Can you please wait for a while because there is some interruption.
Oh, Mahendra, just switch off the camera. Mahendra, switch off the camera. Mahendra, switch off your camera. The camera off பண்ணிடுங்க. Camera off பண்ணிட்டு ஒரு audio மட்டும் பண்ணுங்க. கேமரா ஆஃப் பண்ணிட்டு ஆடியோ மட்டும் பண்ணுங்க. ஆடியோ.
Yes.
கேமரா ஆஃப் பண்ணிட்டு வெறும் ஆடியோ மட்டும்.
There is some. Hello. Yeah, okay, can you wait for 2 minutes because I think there is some interruption outside actually from my apartment. I think so that is the reason. I think now I can continue. I think so. Extraction, which is including hello, can you hear me? Hello. Hello. Hello.
Yes, sir.
Switch off the camera, Mahendran. Yeah, just switch off your camera. Hello, Mahendran. This is Dr. Ubaidullah. Camera switch பண்ணிருங்க camera. Only audio மட்டும் வச்சுங்க. நான் பண்றதுக்கு ஒரு நிமிஷம் நான் பண்ணிடுறேன். கேமரா வெப்காம் ஷேரிங் ஆப்ஷன் எடுத்துறேன் நான் ஒரு நிமிஷம்.
Hello.
Just a minute. I'll do it.
Hello. Is it okay now? Hello.
Yeah, just a minute. Wait.
Hello. Yeah. It's including, I think we have so many methods of extraction, including chocolate and this one. Extraction solvent selection is very, very important. So we have plenty of solvents because our aim is to isolate polar compounds. So we need to select the polar solvents as for the extraction process, which including water, ethanol, and methanol. So normally the extraction of hydrophilic compounds uses polar solvents such as methanol, ethanol, and ethyl acetate. So for extraction more lipophilic compounds, we use a dichloromethane or a mix of dichloromethane. So later, what we need to do, after the extraction process, we go for the concentration of the extract, which including by using rotary evaporator. So after the extraction, we go for isolation. So the isolation and identification. So the isolation has been done by column chromatography and flash column chromatography. So after that, the phytochemical screening has been done by qualitative phytochemical analysis, and we also use FTIR as a FTR and ATR to get to know the what are the phytoconstants which are present in the vegetable plants. So after that, we go for a fractionation and isolation. After fractionation and isolation has been done by chromatography and flash column chromatography techniques. So after that, we go for identification and characterization of isolated compounds. So normally we do isolation, identification, and characterization by spectroscopic techniques. I think there is some interruption. I think now will be okay. Hello. Is it okay? Is it audible, my voice? Hello? Hello?
It is getting better, sir.
I think now...
Earlier there.
Yes, sir, audible.
Actually, outside my apartment, they are doing fogging. So that is why there was a sudden sound came. So sorry for the interruption. I think now it will be okay. So after the identification and characterization of isolated compound, we go for in vitro and in vivo studies. So in vitro studies, which including antimicrobial, antioxidant, cytotoxicity studies, we do a lot of in vitro studies. Then based on the in vitro studies, we take the potent compounds into the in vivo studies. So that is what we normally do in the basic research. So this is whatever I have been explained so far, what we are currently practice and what we do in our current research. So this is considered as a drug discovery and development process. So normally in the drug discovery and development process, hundreds of thousands of molecules are tested every day, and the cost of the screening is very expensive, and discovering new drugs is becoming more complex and time consuming as well. So what we need to do, we need to come up with the innovative ideas to optimize all the steps of development of new drugs, especially drugs that are not. So what I'm going to tell about now about the base, how we can convert the basic research in natural product convert into innovation in product. So I'm going to highlight 3 products that have been done by my research students. So this one, the first one is fingerprint powder, which is obtained from natural source. So fingerprint powder is mainly used to visualize the fingerprint in the crime scene. So the later fingerprint visualization chemicals are toxic and potential hazardous. So what we have done, we come up with a new product. That is a Nanoparticle plant seed powder that has been used as a fingerprint identification. So seeds of durian. Durian is one of the main plant materials which is available in Malaysia, and the seed which has been used as a latent fingerprint powder analysis. The durian seeds are made to be a powder form for the development of cheaper and more effective latent fingerprint powder for better visualization. So what we recommended, the latent fingerprint powder which is obtained from the seeds have better visualization when compared to the existing fingerprint powders. So it is easily available and cheap, and it is one of the green technology and non-toxic and safe. So the second another innovative product is natural ultrasound gel. So actually normally ultrasound gel plays a major role when you take ultrasound machine. So it is one of the conducting medium between the tight bond and the skin of the probe. So the demand for ultrasound gel is worldwide, because ultrasound gel can be used to take ultrasound images as well as in the ECG recording. So normally they use ultrasound gel which is made up of synthetic chemicals and polymers. So now what we have done in the natural product, we come up with alternative solutions or something. So we made up with this ultrasound gel by using seed powder. So that seed powder containing some kind of starch, and even the starch powder can be used as an alternative to make as an ultrasound gel. And this given very better resolution and compared to the other products. So we recommended these techniques to the physicians, radiologists, and physiotherapists to get better results when they use ultrasound machine. So another product is red cabbage as pH indicator. So normally red cabbage, which is containing anthocyanin, that can change color depending on the acidity of its environment. So this has been made it into a alternative to test the pH of any kind of solutions. So this cabbage solution will give different color based on the pH of the particular solution that you are going to test. So next one is a pharmacy related innovative product, that is encapsulated soft gelatin capsule for pharmaceutical and nutraceutical applications. So it is a combination of trigona honey and olive oil and black seed oil. And another one is from seaweed or cocktail shell powder for bone tissue engineering. So normally for bone tissue engineering, we use some kind of synthetic chemicals and also the non-biodegradable products. So this has been produced by seaweeds and cocktail shell powder and water. So this is biodegradable and non-toxic as well. I think now the sound is better because the outside they are doing fogging. That is the reason this sound has come. So sorry for the interruption. And the next product is mobile antibiotic sensitivity testing. So normally when we do antibiotic testing, so we need to check the zone of inhibition. So normally when we do zone of inhibition, we use normal vernier scale to check the zone of inhibition. But this is one of the automatic machine which can measure the inhibition of zone and interpret the result automatically. So there is not much manual work. So once you've done the antibacterial studies, once you get a zone of inhibition, that is not necessary to measure by manually. So you can keep this disk into the machine, so automatically that can detect what is the zone of inhibition, and that also automatically compare with the standard antibiotics. So next one is rapid detection of methicillin and vancomycin resistant staphylococcus areas by a newly developed cold chain free PCR assay. By using this one, we can high sensitivity, specificity, and lower detection. This kit can be used as in ordinary hospital labs. So this we can detect the vancomycin resistant staphylococcus areas and methicillin resistant staphylococcus areas. And this everybody knows. I think this product has been already developed and it is in the market. So smart vein locator with the depth and thickness information. So normally visualization of vein also very difficult previously. And now there is an innovative product, so we can visualize the vein which is present in the skin, especially the person who is having darker skin tone and presence of scar, black spot or hair and thick subcutaneous fat. So this product can visualize the subcutaneous vein and especially efficiently localize the veins and its position. So next product is TB NanoSense Kit Test Kit. So this NanoSense Test Kit can be used to detect Mycobacterium tuberculosis. So that is what I told you, we need to have some kind of collaboration. So if you work only with the pharmacy department and pharmacy people, so we may get idea. But the idea, if you want to implement it, be an innovative product, and if you want to go for commercialization of those products, definitely we need to have a collaboration with some kind of technology-based people, like engineering people. So whatever the idea you tell, so definitely they can come up with a final product which can be useful to the society. And there are oral HPV 16 lamp kit. It can be used to detect HPV 16 and home care uric acid test reader, which is available in the market nowadays. So this kind of innovative product only we need to ask our students to come up with some kind of idea to overcome the disadvantages of the existing products. So we can come up with a new product with the technology and easily we can detect all kind of this one. Then rapid test kit and diagnosis for leptospirosis. So leptospirosis is one of the infections which are mainly from the animals to human. So this is very common in Malaysia and some Asian countries. So previously to detect this leptospirosis, it takes a lot of time actually. It takes 1 day or 2 days to get know the result whether the person is infected by leptospirosis. And now we have the test kit which can get the result within 15 minutes of time. And another one, dengue fever test kit. So normally in dengue fever test kit also is available nowadays and we can get the result within 10 minutes of time. And this is tea honey generally is normally this honey is was obtained from Indonesia and they are using for to reduce nausea and vomiting in pregnant women, because normally to avoid nausea and vomiting normally they use some kind of tablets. So that can be avoided when they started off using this kind of honey, and this product also available in the market. And some innovators even they come up with cookies which is made up of egg cells and Moringa oleifera, because Moringa oleifera we know it is a healthy leaf which can be used for many medicinal purposes. So if some people they try to avoid to take some kind of leaves as a medicine. So what the innovator they thought they come up with the cookies which is made up of these egg cells and leaves. So then even the innovator they come up with the chocolate also nowadays with some medicinal plant which can be used. And this is a herbal ice cream which has numerous medicinal properties including antioxidant and anti-diabetic. And this one is very interesting product which has been come up with one of our innovator. So normally the pillows are made up of with some kind of cotton, but this pillow is special pillow which is made up of some Vitex negrundo leaves, which is also available in India. And this will give some kind of peacefulness to when the people sleep with this pillow. So what the innovator they come up this pillow together with the music and massager, which makes it is a comfortable, relaxing pillow. And another product is this is a Corsa gel, which is mainly made up of corn silk. So normally what we do, the corn powder or corn starch can be used for many purposes, but the corn silk normally it is one of the waste product actually. So how we can come up the product from waste to an innovative product. So this is one of the innovative product which is made up of corn silk, and this can be used to treat especially for the diabetic foot ulcers. And next one is mosquito repellent liquid, which is made up of Plectranthus ambitionis. So it is very effective and safe and affordable. So normally the mosquito repellent liquid which is available in the market, that is made up of synthetic chemicals. So this one is something which is containing natural ingredients. So this way we can think some imagine. This is a very simple product. Even we can use any kind of natural product which can be used as a mosquito repellent. So that can be converted into a plant liquid. So you can use some kind of oil which can be obtained from the plant material as one of the ingredient in this kind of product. So this also can be commercialized easily. And the mosquito repellent spray, this also made up of with a natural product. And another one is Cevix Randu honey to treat cervical cancer, which is containing quercetin, luteolin as a natural compounds. And another interesting product is food freshness sensing device. So that way normally we get many ideas. It is not necessary the pharmacy means we need to go for innovation something related only to the medicines. So we can go for something like this kind of product also. So this product can detect the gas which is released from the contaminated food and alert the user, which will prevent food poisoning. And another one is Nochi patch. So normally it is made up of using leaves and flowers, and it can be used as a head patch or as inhalant in hot water. So this has been used as a patch. So normally, for example, if you select Nochi leaves as a liquid form or maybe the extraction form or maybe some kind of other formulations, definitely the people, they don't like to use it. So what innovators did, so they come up with a patch type. So what the patch is containing this Nochi leaves ingredients. So normally that can be used to treat headache and sinus problem. And another product is hand sanitizer, which is made up of with basil leaves. It can fight against bacteria and virus. And wound healing ointment, which is made up of Bromelia odorata leaves. And this one is natural healthy sugar from papaya. So normally we use normal sugar for all kind of purposes. So this has been made up of with papaya. So the papaya extract has been concentrated and has been converted into a solid brown sugar, and this can be natural sweetener. And this product is come up with from the waste material. So normally we take banana and we throw the banana peels. So this banana peels also can be used as one of the innovative products to remove scars and skin. So this product also even available in some Asian countries. It may not be available in India, but Asian countries like Indonesia, they have this kind of product, which is made up with banana skin. And Evapa gel, this is made up with rambutan peels, which can be used to prevent dental plaque on teeth. And this is one of the very interesting products is medicated eyeliners, which is containing ophthalmic medications, which is inside. This is not a cosmetic eyeliner. So this is a medicated eyeliner, which is containing some medicine, especially for the ophthalmic purposes. So this can reduce the risk of infections. So next, I'm going to highlight what are the innovations in cosmetic products, because we know cosmetic products we can make it easily by natural source. And many beauty products are designated for applying for face and body. And we know the cosmetic product is the market is very big actually. So normally nutraceutical product, the market size is the market value is 175 billion in 2018. But the same, if you check with the cosmetic product, it is 700 billion US dollars actually. So that means we are spending more money for the cosmetic products. But what are the cosmetic products which is available currently in the market? Mostly it is made up of synthetic chemicals. So what we can do, we can come up with something made up of natural products. So that is what I'm going to highlight after this. So this is one of the body soap, which is made up of citrus passion, so that are suitable for all age group, and it consists of citrus leaf oil. It is claimed to be used as cancer-related skin infection. So this, especially this soap, is mainly used in the market. It's not like something like innovative type. So normally they sell it up cream, ointment like that. So this is something new, which is they come up with the iPhone pack, which is containing some natural ingredients, including tea powder, peel cucumber, carrot, and aloe vera gel powder. And next one is the healing herbs cream, which is made up of aloe vera, guava leaves, turmeric, and neem leaves, which can shorten the healing days and have strong antibacterial properties. Because normally when you make any kind of products, especially natural-based product for cosmetic and for the other kind of antibacterial or antimicrobial properties, so we need to have antioxidant and anti-inflammatory properties as well. So that will be more useful to the activity. So that is why when you choose the plant material to make this kind of product, the very important thing is the plant material must have antimicrobial properties, analgesic and anti-inflammatory properties, as well as some other properties. So that kind of plant material can be easily
muted.
Especially the mask type, which is containing only one ingredient or mixture of ingredients in all the places. So the beauty of this product is, so they get different ingredients in different part of the this mask. So this can be used for many skin problems, or maybe this can be used to glow up skin. So this is one of the very recent innovative product, that is Microneedle Patches, so which is containing hyaluronic
muted.
Cosmetic product, when you look at into the market, so that is retinol is one of the active ingredients which is mainly present. Retinol means that is vitamin A normally, but so nowadays the innovators, they come up with a new compound that is mainly obtained from the plant material, that is Glucacia. It is mainly obtained from Morelli Kumaraswamy. So this product, this compound has been replaced for retinol, because retinol having more disadvantages, especially retinol cannot be used for all skin types, because that is allergic to most of skin types. So that is why now they changed to Bocatiol. Bocatiol is one of the active ingredients which is present in the plant material. So this has been nowadays many products. When you look at the product, Bocatiol is one of the active ingredients which is present in the cosmetic product. So and another great thing about the ingredient is it is suitable for most skin types and has benefits that extend far beyond a naval glow. Next will be CBD oil, which is mainly obtained from cannabis. So that also used as one of the active ingredients for many cosmetic products. Especially this can be used to treat dry skin, psoriasis, and eczema. So the inflammation in the root of all evil, whatever that the signs of aging. So this product is also this CBD oil is one of the main ingredients nowadays. It is available. It is obtained from cannabis sativa. And normally primary and beta and alpha hydroxy acid, glucamic acid has been used for cosmetic products, and now polyhydroxy acid, which is mainly obtained from apricot. So this has been used as one of the active ingredients in the cosmetic product formulations. So next I'll be highlighting, I think, so sorry for the interruption, because something outside my apartment, so I cannot do anything. So next will be about the medicinal plants in cosmetic formulations. So I'm going to explain, I'm going to highlight some important medicinal plants. Maybe if the future researchers can use this plant material as one of the ingredients for the cosmetic formulations, and those products can be commercialized very easily. So I'm going to highlight some important medicinal plants, including aloe vera. This is aloe vera is mainly used in lipstick and hand cream, facial cream, which is containing antioxidant, antimicrobial, and anti-inflammatory properties. So we know aloe vera has been used for many medicinal purposes, but as a researchers, we do a lot of research, but we don't do more something like innovative products, because once you come up with an innovative product, then only that innovative product can be used by the customers. If you do research, research cannot be used by the public actually. So that is what we need to practice with our students. And next one only kurkumalanga, turmeric is containing curcumin as one of the main ingredient, which is more beneficial in reducing unnecessary facial growth. So this also can be used for many cosmetic formulation in the future. If you want to do some innovative product on, especially on cosmetic products. The next one is this one. Coconut oil has been used as a excellent ingredient for the hair cosmetics. And cucumber is effective in cooling and healing and soothing of skin irritation from sun exposure, skin problem, etc. So this also can be used. That is, we have some common medicinal plants which is very easily to get it all over the places. So those kind of ingredients only you need to select. But if you select some ingredients, especially the medicinal plants, which is not available easily to get it, so definitely you cannot make a product. So you have to select the material which is available easily and nearby to your places, so that you can easily make up the cosmetic product. Next one is this one is tea tree oil. So that also has been used to make a lip balm or lipstick, which can be used to treat sore or cracked lips. And this one pomegranate. Pomegranate peels are used for many cosmetic formulations, and this is containing some antioxidants which can be used for skin whitening and skin repairing. And papaya, we know papaya has been used for many medicinal purposes, including antioxidant, antibacterial, and anti-inflammatory purpose. But we don't have much product. We have some product in the market. But what innovators, what we need to do, we need to make a product something innovative, not like creams or ointments. So once you come up with a new idea, how that ingredients to be converted into innovative type, something like what I have been mentioned, the mask type, or maybe the miracle icon type. So something like that we need to think. So those kind of product only very attractive by the customers. And the next one is of mangosteen. So it's one of the ingredients in the hair growth and hair tanning products. And hibiscus, as we know, this has been used for long ago with many I think especially the village people, they normally use it for increasing the hair volume and slowing down the graying hair. But we don't have much product based on this hibiscus process analysis. So in future, if you have any plan to come up to make some kind of cosmetic product based on natural products, so you can include these kind of ingredients. So these ingredients will be very, very useful, especially if you want to make some product which is related to hair growth or to increase the hair volume, something like that. Then another one is henna. You know, henna is opted to be incorporated in cosmetic products due to wound healing and skin repairing activities. So these are the main important medicinal plants that have been used for cosmetic purposes. That is what I highlighted on this plant material. So in future, if somebody, for example, if the student want to make some kind of innovative product, they can use this kind of ingredients. These are very easily available medicinal plants in all over the places. So you need to choose the plant material, something which is easily available nearby your places, so that the innovation will be very, very easy, and making the product also, it will be very easy. And coming to the conclusion, so innovations using natural products play a major role for entrepreneurship in pharmacy practice or education, and will assist in establishing pharmacist entrepreneurs. So our main aim is we need to produce pharmacist entrepreneurs. So how we can produce pharmacist entrepreneurs? So they need to have some kind of innovative ideas. They must do some kind of innovation during when they do bachelor program or master program. So then that idea, based on that idea, they will get some kind of innovative mindset after completion of their course. So it is not necessary they need to bid for a job. So they can open a small scale industry. They can prepare this kind of product. They can get approval from appropriate agencies, and they can sell it up. So that this will be very, very useful actually. So my final word to the faculty members of pharmacy who are interested in incorporating entrepreneurship education in schools or colleges of pharmacy and implement this innovative culture among students. So we need to start implementing innovation culture, because we do a lot of research, we do a lot of publications, but finally that cannot be converted into a product. So we cannot see the product from the publications. But research also very, very important, because research will give some idea to make some innovative product. And we know that natural products are playing a major role in all kind of innovative products because natural products are safe and toxic free when compared to some synthetic products. So that is why people they adopt with natural products. And this kind of innovation, I think definitely the student in future, they are not necessary to search for job. So they can create job and they can give more opportunities to the people and all the other young pharmacists to make some product, especially they can make some cosmetic products based on the natural product as one of the ingredients. So with that, I would like to thank very much for the opportunity and thank you very much for listening. I'm really sorry for the interruption happened because outside my apartment, I think they're doing fogging. So I think that is why suddenly the sound has come, so I cannot avoid that one. I'm really sorry for that. Sorry for the interruption. So if anything, maybe I can share my slide to the organizer. Maybe you can go through and if you have any questions or anything in future, maybe you can contact. And we are more looking for collaboration also to make some kind of innovative products. And I given my contact number and email ID, so you can contact me at any time. So definitely we can discuss with some kind of innovations and we can bring some product into the market. And we can make realistic whatever we do research. Because normally when we do research, we cannot see the final outcome of research because we do only publications, and that can be useful for the promotion and other things for the faculty members. But for the student side, if you come up with some product, you can proudly say that is your technology, your product, something like that. So that is what we are practicing over here with our students. I think this session will be might be very useful for every one of you. I think that's all from me, and thank you very much once again. And I will hand over to the organizer for the question answer session.
Thank you, sir. Thank you. It's good to see a lot of products, a lot of things we see naturally coming the good products out of that. Now I call upon Ms. Sheila, madam, to post questions if there is to the presenter.
Yes, sir. Thanks for the wonderful session, sir. There are some questions from participant side. Is there any herbal transdermal patches in market? Sir?
Yeah. Hello.
Am I audible? Is there any herbal transdermal patches in the market?
Hello. Can you repeat your question? So I couldn't hear voice.
Is there any herbal transdermal patches available in market?
Yeah, transdermal patches, I think it is available in the market. That is why what we need to do, there are many products which are available already in the market. So what we need to do with something new ingredients for the new medicinal purpose, for other kind of infections or maybe diseases or some kind of skin problems like that. So it is not like that if there is a transdermal patches available in the market, it is not necessary. It's not like you should not do the innovation based on that one. So there are products are available, but that are existing products. So you must come up with what is the disadvantages of those products. So that will give more innovative ideas to go for the new innovative transdermal patches. Yeah.
Okay, sir. Even though herbal plants are safe, but still those fixation of herbal drugs not succeed. Can you suggest some ideas how to achieve this? Suggestion for both fixation in herbs.
Yeah. So most of the time we used to talk natural products are safe, but all the natural products are not safe as well. There are certain natural products and plant materials which are highly toxic when compared to the synthetic chemicals. So what we need to do, where we are like is we are not doing proper standardization. So we need to standardize what is the active ingredients which are present in the particular plant material. So when you go for the isolated compound or something like bucachiol or retinol, something like that. So that kind of product, maybe you can go for toxic profile testing, and that will be easy to bring those kind of products compared to the plant extract. Because plant extract, it will be the constituents will be vary from all the time. So for example, if you collect the plant material from one area to another area, definitely there will be a difference in the ingredient side. So we don't know exactly the quantification of each ingredient which is present in the product material. So that way, it is very difficult task actually to come up with a product with a natural product. But that way we need to do more standardization, especially what is the active constants which are present in the plant material. So that way only we can bring the products into the market. Otherwise, if you don't do any standardization, so it is very hard and very difficult to convey the customers, as well as it is very difficult to get approval as well for the cosmetic products into this one. Yeah.
Okay, sir. Whether herbal tech researchers are showing interest in clinical trials or ending their research till animal studies? Tell me.
Yeah, normally some cosmetic products, it is there is no standard or developed animal studies model, as well as clinical trials also not available for those kind of products, especially for the cosmetic products and something else. So if you your innovative idea is something related to pharmaceutical, something like medicine side, internal medicines, so then definitely we should go for clinical trials and animal studies. So for the cosmetic products, normally we test intensity test and whether this any kind of, for example, if you prepare any cosmetic products, we test with the human volunteers, healthy volunteers, to check whether this product is giving any kind of irritancy or skin redness to the person who is tested with this kind of cosmetic product. So normally we don't have much clinical trials and animal models, especially for the cosmetic products. But for other medicines, definitely we should go for animal studies and clinical studies before we bring into the market. Especially I mentioned medicated this one medicated eyeliner, right? So what are the ingredients we include? That is already prepared by the innovators, but they put that the ingredients is one of the already existing products which can be used for ophthalmic problems. But if you want to use some natural things, then definitely we need to go for a clinical trial. So if for the external purpose, something like if you want to make creams or ointments or cosmetic products, so it is not necessary that you need to go for clinical trials. So that's all.
Okay, sir. Last question. Is there any herbal
Muted.
We need to induce the cancer to the animals, then later the skin to be tested with this, what are the products that you have been prepared. So, but commercially it is not available, which is especially made up of natural products. That is why we innovators, we need to create more innovators to come up with those kind of products. Once you get an idea, you just think what the medicinal plant extract or maybe the ingredients to be added to those products, then try to implement and do proper testing for those things. Then that can be commercialized. Otherwise, it is very difficult task actually to bring those products, especially for the skincare, especially because you are mentioning that can be that product to be used for skin infections related to cancer. So that means proper test is supposed to be done, and including the even the clinical trials as well.
Okay, so
Mute.
Purushot Prabhu sir to give vote of thanks.
Okay, sir. Good evening to all the faculties and students from various colleges throughout India. First, I wholeheartedly thank our management for the support given to conduct the webinar 5 series in advanced scientific knowledge in times of pandemic. I would like to thank our beloved principal, Dr. Grace Ratnam ma'am, for giving constant support and encouragement for doing this webinar. I would like to thank our resource person, speaker Dr. Morelli Kumaraswamy and Dr. Mahendran Segar, who accepted immediately to speak on our webinar 5 series. Both the speakers were done excellent and interesting presentation, which will motivate our students to take up the pharmaceutical research in nanomedicine and natural product research. I thank my departmental staff who given support to make this webinar success. I would like to take this opportunity to thank our webinar team, Dr. Ubaidullah sir, Vijay Kumar sir, Sivakumar, for enormous support to conduct this webinar. Last but not least, I thank all the faculties and students from different colleges for their patient listening and participation in our webinar 5 series. Once again, I thank everyone. So we will soon meet in next webinar. Thank you.
Thank you, sir. We are planning for next webinar too. We'll come to you, let you soon confirm the date, and we'll share the registration link. Thank you.
Yes.
Thank you,
Thank
sir.
you. Thank you, Murli and Mahendran sir.
Yeah, thank you, sir. Thank you so much.
Thank you, Murli. Thank you, Mahendran.